ठीक है. So today's topic is C3 and C4 pathways of carbon fixation. And we all know that the carbon fixation process is nothing but the photosynthesis process, right? Where the first reaction, where the sunlight is utilized, it is called as a light reaction. So firstly, we will go to light reaction. So light reaction is the reaction where there is synthesis of ATP in the presence of light. Similarly, it may produce a reducing energy. From NADP, it produces NADPH. This is reducing energy. This may be produced or may not be produced. Similarly, there is evolution of oxygen, but again, it may be evolved or may not be evolved. So, synthesis of ATP is present in both the light reactions. So, there are two types of light reactions: cyclic light reaction and non-cyclic light reaction, or cyclic photophosphorylation and non-cyclic photophosphorylation. Phosphorylation. What is phosphorylation? ATP synthesis, where the phosphate, inorganic phosphate, is added to ADP and there is production of ATP. So addition of inorganic phosphate is called as phosphorylation. Now this phosphorylation, which happens in photosynthesis, it occurs in presence of light, so it is called as photophosphorylation. We have another process of phosphorylation which occurs in presence of oxygen in mitochondria. मायटोकॉन्ड्रियामध्ये पण एटीपी सिंथेसिस होतो. ज्याच्यामध्ये ऑक्सिजनच्या प्रेझेन्समध्ये हीच प्रोसेस कंप्लीट होते. त्या प्रोसेसला आपण काय म्हणतो? ऑक्सिडेटिव्ह फोटोफॉस्फोरायलेशन. ही फोटोफॉस्फोरायलेशन वेगळी आहे आणि फोटोसिंथेसिस मधली फोटोफॉस्फोरायलेशन वेगळी आहे. सो वी विल डील ओनली विथ फोटोफॉस्फोरायलेशन. So cyclic photophosphorylation. Cyclic photophosphorylation, it occurs in stroma thylakoid. It occurs in stroma thylakoid. We have seen the structure of chloroplast. It includes grana thylakoids, and these grana are connected by stroma thylakoids. The process of cyclic photophosphorylation occurs in stroma thylakoids. It may or may not occur in grana thylakoid. So mainly it is present in stroma thylakoid. Now in this reaction, there is a photosystem 1 which is involved in the process. This photosystem 1 has a reaction center which is called as 700. It is controlled by chlorophyll A molecule and it can absorb the light wavelengths up to 700 nanometers. 700 नॅनोमीटर पर्यंतचे वेव्हलेंथ हे ऍब्सॉर्ब करू शकतं, म्हणून आपण याला रिएक्शन सेंटर 700 म्हटलं आणि त्यामुळे जी तयार झालेली फोटोसिस्टम आहे याला आपण म्हणतो फोटोसिस्टम 1. मगाशी जसं सांगितलं तसं या फोटोसिस्टम मध्ये आहे रिएक्शन सेंटर प्लस ॲक्सेसरी पिगमेंट्स, अँटिनाज आर देअर and together they are working as a photosystem 1. This photosystem 1 is associated with number of electron transport molecules. त्याच्याबरोबर काही इलेक्ट्रॉन कॅरियर असोसिएट झालेले आहेत. हे इलेक्ट्रॉन कॅरियर्स एक्साइट झालेल्या इलेक्ट्रॉनला ट्रान्सफर करतात आणि या प्रोसेसमध्ये एक एटीपी सिंथेसिस केला जातो. when the electron passes through a complex called as cytochrome B6F complex. या कॉम्प्लेक्स मध्ये एक प्रोटॉन पंप केला जातो. आता हे जे मेम्ब्रेन आहे या मेम्ब्रेनची एक बाजू आहे स्ट्रोमा, म्हणजे ओपन असणारी बाजू, वरची किंवा खालची बाजू आपण म्हणूया. आणि या थायलॉइडच्या आतमध्ये इथं ल्युमेन आहे. ओके, सो दिस ल्युमेन इज प्रेझेंट हिअर. इथं आपण ल्युमेन दाखवलंय, म्हणजे याच्या खालच्या बाजूला अजून असंच मेम्ब्रेन असणार आहे ज्याच्यावरती फोटोसिस्टम असते. नाउ दीज फोटोसिस्टम्स दे हार्वेस्ट लाईट एनर्जी. याच्यामधून लाईट एनर्जी कॅप्चर केली जाईल. दिस लाईट एनर्जी विल excite the electron present in this photosystem. That electron is donated by the reaction center chlorophyll A. As soon as it is donated, it is captured by an unknown molecule. So here one unknown molecule is there, which is the first molecule which captures the electron, and then this unknown molecule transfers it to the ferrodoxin. हा unknown complex जो कोणी आहे तो molecule electron acceptor आहे, and then this electron is given to the ferrodoxin. So here we can see the excited electron is given to the primary acceptor and then to the ferrodoxin. Ferrodoxin passes this electron down to the energy stream to the plastoquinone, then it transfer to cytochrome complex, cytochrome B6F complex. It comes to plastocyanin, and finally again it returns back to the reaction center. This cyclic movement of electron is there. So whatever electrons are emitted, those come back to the reaction center. So this process is called as a cyclic photophosphorylation. यांना आपण cyclic photophosphorylation असं म्हणतो, because the electron which is given out by the reaction center, it comes back to the reaction center. Okay. Now we can see the second part, ATP synthesis. आता ATP synthesis कसं होते ते आपण बघूया reaction center मध्ये. When the electron passes through the cytochrome B6F complex, it pumps one proton from the stroma to the lumen. स्ट्रोमा मधून एक प्रोटॉन या इलेक्ट्रॉन बरोबर कॅरी केला जातो आणि तो ल्युमेन मध्ये पंप इन केला जातो. So in this way, the number of protons H plus ions are accumulated, concentrated in the lumen. And whenever these H plus ions goes through ATP synthetase complex, हा शेजारी दिसतोय तो आपला ATP synthetase complex आहे. याच्यातून ज्या वेळेला प्रोटॉन पंप आउट होईल त्यावेळेला ATP synthesis होईल. So addition of proton to the lumen is equivalent to addition of synthesis of ATP. Okay, so this reaction which is coupled with ATP synthetase molecule, it produces ATP. So in this process, when two electrons are emitted, one ATP molecule is synthesized. Now here we can see there is a no splitting of water. Okay, so this reaction produces only ATP. There is no photolysis of water, no generation of oxygen, no production of reduction power. Now coming towards the non-cyclic phosphorylation. This process, it happens in grana thylakoid. It happens in grana thylakoid. So again, I will draw the chloroplast. This is the chloroplast double membrane system. There are number of granas, and these granas are connected with each other by stroma thylakoid. So it happens in grana thylakoid. On this grana thylakoid, we have both photosystems which are closely associated with each other, photosystem 2 and photosystem 1. Both have the reaction center chlorophyll A. फक्त फोटोसिस्टम 1 मधलं क्लोरोफिल ए जे आहे ते 700 मॉलिक्यूल आहे. इन फोटोसिस्टम बी इट इज 680. या दोन्ही सिस्टम्स एकत्र काम करतात. And in this process the ATP is produced, NADPH, that is reducing power, is produced, and water molecule is photolyzed. Water molecule is photolyzed to produce oxygen. How it happens? Let us see. So this is the organization. Note again, this is the membrane. Outside this membrane there is a stroma. आतल्या बाजूला we have a lumen, and this lumen is present in this grana. या ग्रानाच्या आतमधलं जे ल्युमेन आहे त्या ल्युमेनच्या बाबतीमधला आपण विचार करतोय आता. So this lumen is involved here. Okay, there are two photosystems, photosystem 2, photosystem 1. Both have accessory pigments, both have reaction centers. Photosystem 1 has a reaction center 700. Photosystem 2 has a reaction center P680. Now this reaction starts with photosystem 2. So whenever photosystem 2 receives the photons, light energy, this is also represented as H mu, which is utilized for photolysis of the water molecule, as well as the electron which are excited from this reaction center, they will be given out. The first electron acceptor will, this is again unknown, will accept the electron and it will be transferred to the chain. Plastoquinone will be there, it will be reduced to plastoquinone. Then the electron transferred to cytochrome B6F complex, then it comes to plastocyanin, and further these electrons are transferred to photosystem 1. So those electrons which are coming from photosystem 2 are now reached to photosystem 1. Now here these two electrons are further excited by absorption of light, and these two electrons are given to ferrodoxin. In presence of enzyme ferrodoxin reductase, the ferrodoxin get reduced, and finally these electrons are given to the NADP, where NADP is converted to NADPH. And the final acceptor of the electron is NADP. Okay, so this is the final electron acceptor. Final electron acceptor. This final electron acceptor will utilize these electrons plus photon to produce a reducing energy called as NADPH. Okay, one side is clear. Now, as this electron which is given by photosystem 2 does not come back, it is given out permanently. This process is called as a non-cyclic. So, how this cavity or the vacancy of electron is fulfilled? So, that is filled up by another process which is called as a photolysis of water. या प्रोसेससाठी जे इलेक्ट्रॉन येणार आहेत तर इलेक्ट्रॉन या फोटोलिसिस ऑफ वॉटर मध्ये येतील. दिस एनर्जी विच इज ऍब्सॉर्ब बाय द फोटोसिस्टम 2, this energy is also transferred to water molecule, where the water molecule is split down into two molecules, H plus and electrons plus oxygen. Okay. So this is a reaction which is controlled by one complex. This is called as oxygen evolving complex. Okay, so this is oxygen evolving complex, OEC. It is dependent on manganese and calcium. So this photolysis of water occurs like this one. Two water molecules will produce one oxygen molecule. This will be evolved. Four photons will be released. By ionization, they will produce four photons plus four electrons. याच्यापासून electrons तयार होतील. These electrons will go to photosystem 1 and then later on those will be transferred to electron transport chain for non-cyclic photophosphorylation. These photons get accumulated in the lumen. And finally, when these photons are pumped through ATP synthetase complex, they will produce ATP. This is the overall reaction. Here the sunlight and water molecules are utilized and oxygen is the byproduct, ATP is the byproduct, and NADPH is the byproduct. So this is the light reaction which does not involve any relation with CO2. So this is independent of CO2. Now the CO2 is fixed in another process which is called as a dark reaction. आपण बघितलं लाईट रिएक्शन मध्ये NADPH तयार होतो, ATP तयार होतो आणि ऑक्सिजन प्रोड्यूस केला जातो. आता CO2 फिक्सेशन कसं होतंय ते आपण बघणार आहोत. So this process is called as a dark reaction. So dark